Hello, and welcome back, fellow SPCH Rangers. I'm Linda, and today I'd like you guys to quickly imagine that what if I told you it wasn't really me giving those speeches in the first two videos, that the Lindas you saw talking about snowboarding and scuba diving were really my clones. Well, that is just an example of one of the limitless scenarios on how great having your own clones can be. At the end of my speech, you will agree that supporting human cloning initiatives will greatly benefit mankind, not just to do our SPCH 100 homework for us, but according to the National Institutes of Health, a major branch under the U.S. Department of Health and Human Services, medical advancements in human cloning technology, such as stem cell research, can be used to screen and test new drugs, find new cures for currently terminal diseases, and ultimately saving human lives. And that is just the tip of the iceberg. I like to ask how many of you guys know someone born with a genetic disorder such as muscular dystrophy, where they can barely walk or feed themselves, cystic fibrosis, where they must rely on a ventilator to breathe because of degenerative lung function. If only there was a way we could prevent another human baby ever being born with a known genetic disorder. How many of you know someone who has been diagnosed, or have hoped personally yourself will never be diagnosed, with a chronic and progressive debilitating disease such as Parkinson's disease, where your neurological faculties cause you to suffer uncontrollable tremors, or multiple sclerosis, whereby your central nervous system components and spinal cord slowly break down on you? How many of you know someone who has died due to a terminal disease, such as those in the last stages of Alzheimer's or stage 4 leukemia? Or perhaps you have a family member or a dear friend who recently lost a loved one due to heart, kidney, or liver failure? If only we possessed the knowledge to make the medical breakthroughs that would save their lives. Well, all the questions I just proposed to you are just a handful of the reasons why further advanced research into human cloning technology is needed. When scientists use the term cloning, they mean something very different than what the public views cloning as, thanks to the plethora of science fiction movies out there. We can't just step into a machine and walk out with a clone, like in Hollywood movie magic style. But scientists around the world have identified the conditions that would allow stem cells to be grown and genetically reprogrammed in the laboratory through techniques such as somatic cell nuclear transfer. The American Medical Association refers to this process as therapeutic cloning. In this emerging scientific field of genetic engineering, advancement in human cloning technology is fully dependent on stem cell research. Stem cell research ultimately holds the key in discovering the desperately needed medical breakthroughs to find cures to these currently aforementioned incurable human conditions. So, what are stem cells? Well, according to an article in the American Medical Association, stem cells differ from other kinds of cells in the body as they are a specialized type of cell that contain the genetic info in the creation of all our vital organs and tissues. Scientists primarily work with two kinds of stem cells: embryonic stem cells and somatic or adult stem cells. All stem cells, regardless of their source, give rise to specialized types, such as heart muscle cells, which contain the genetically encoded DNA sequence instructions to pump blood through our body, or red blood cells, which contain the encoding on how to carry oxygen molecules through our bloodstream. In bone marrow and muscle tissue, adult stem cells contain the encoded instructions to generate replacement cells depleted from everyday normal wear and tear. Given their extremely unique regenerative abilities, stem cells offer new and promising potentials for people suffering from a myriad of terminal diagnoses by replacing the damaged cells in any organ or tissue. Another distinct advantage of therapeutic cloning is that there is no risk of your body rejecting it, as you're using your own genetic material. And that is one of the major obstacles of organ transplantation, compounded by the fact that the number of people needing a transplant far exceeds the number of organs available for transplantation. According to the National Heart, Lung, and Blood Institute, approximately 3,000 people in the United States are on the waiting list for a heart transplant on any given day. According to the National Marrow Donor Program, 70% of patients in need of bone marrow transplant do not have a suitable donor in their family. Human cloning technology and stem cell research offers us the possibility of having a renewable source that we can harvest to replace these vital organs that have been diseased or defective, whether through birth, illness, or accidental injuries later on in life. Just imagine how we can dramatically, if not completely, reduce the wait time for organ transplants, as well as the immunological concerns associated with the procedure. Cloning technologies may also very well play a major role in saving endangered species such as polar bears, which according to studies by the National Geographic indicate that 2/3 of the world's polar bear population could vanish by the year 2050. Cloning technologies may also become a viable tool for reviving already extinct species by resurrecting them from frozen tissue. The successful cloning of Dolly the sheep in Edinburgh, Scotland in 1996 cracked open Pandora's box for cloning animal and human cloning initiatives around the globe. In 2009, scientists from the Center of Food Technology and Research in northern Spain announced the cloning of the Pyrenean ibex, a species of wild mountain goat, which was officially declared extinct in the year 2000. Although the newborn ibex died shortly after birth, it is the first time an extinct animal has ever been cloned, and is why I urge you to say yes to human and animal cloning initiatives. I'd like to leave you guys with a quote from American physicist Dr. Richard Seed, who is one of the leading proponents of human cloning technology, and it goes, "New things of any kind, mechanical, biological, intellectual, always tend to create fear. Don't fear cloning."